So here in 18.2, we're going to talk a bit more about this major histocompatibility complex, or the MHC. So remember that we have already talked about the MHC previously, and we said that this is something that allows our cells to recognize that they are self cells. So now we are going to take a closer look at that and see really how this works. There are actually two different types of classes of the major histocompatibility complex, or the MHC proteins. There is the Mhc Class 1 and the Mhc Class 2. And then they're really just shortened MHC1 and MHC2. So we're going to talk in more detail, but I wanted to see this image first so you could kind of see what we're talking about. And the MHC proteins or molecules are embedded within the plasma membrane. So you can see here our phospholipid bilayer, and these are our self cells that we're talking about here. So for example, in Mhc class 1, we find these in all nucleated body cells. The reason we say nucleated is because red blood cells are not nucleated. They do not have a nucleus. Part of the process of making a red blood cell is that it gets rid of the nucleus toward the end and then becomes a red blood cell that goes around the body. So since it is not a nucleated body cell, it does not have an Mhc class 1 protein on it. Every single other cell in the body does, however. Our red blood cells have other proteins that recognize, or that our body recognizes as self. Not that we're going to talk about that in this class, but they don't have the Mhc proteins on them. So Mhc class 1 we find in all nucleated body cells. This is something that our body, our immune system cells will recognize as self, but it also serves another purpose, and we'll talk about that in just a moment. Our Mhc class 2 over here, so you can tell that they look a little different. That's why they are different. So the Mhc class 1 has some different pieces here. You don't need to know the alpha 1, 2, 3, etc. pieces. Just note that the Mhc class 1 is different than the Mhc class 2. And that the Mhc class 1 is on all nucleated body cells, so that means immune system cells and every other cell, whereas the Mhc class 2 are found on macrophages, dendritic cells, and B cells. But they also have the Mhc1. That's why it's important to note that they're on everything except for the nucleated or non-nucleated cells, which are the red blood cells. And then the Mhc class 2 are only on macrophages, dendritic cells, and B cells. And that's because these are antigen presenting cells. So let's talk about that. We're going to start with our Mhc class 2 molecules, and as I just mentioned, these are on our antigen presenting cells. What that means are these cells that are going around in our body that are able to deal with pathogens, are able to interact with them, are able to recognize a pathogen, most times bring it into the cell, not the B cell so much, but the macrophages and the dendritic cell are able to bring them into the cell because they're phagocytic, and then they can break them down, and then they can present the antigen, which is why it's called an antigen presenting cell. So let's talk about antigen presentation with these molecules. If we look at the illustration over here, we can see that we have this bacterial pathogen, and all of those little red circles on the outside of the bacteria are supposed to be little pieces of antigen. So we could even say epitopes. So those are epitopes on the antigen. So in step 1 here, our bacterium is engulfed by phagocytosis into a dendritic cell or macrophage and is encased in the phagosome. So remember, when we bring in the bacterium, it is in a phagosome. That's this vesicle that contains the bacterium, and then it's going to fuse with our lysosomes. So here we have the lysosomes in part 2 here. We have our lysosomes, and they are going to merge with that phagosome. When they merge with that phagosome, then they're going to digest the bacterium. Remember, that's a phagolysosome, and that's because all of the enzymes in the lysosomes are now going to be breaking down that bacteria, cutting it up into pieces. So you can see that that's what it's doing here. Now the bacteria is no longer this, rod-shaped bacterium. Now it's got all of these little pieces, including the epitopes all over inside this phagolysosome. Then what happens is that the cell is going to take the most antigenic epitopes, so those that are going to produce the largest response, or basically are the things that are least like ourself. Again, looking at those different types of classes, the different biological molecules that can be used to make those epitopes or antigens, likely what they would do would be to process and put a protein antigen there or epitope there. rather than putting something like maybe there's a carbohydrate or a nucleic acid-based epitope. So the ones that are most antigenic are going to be processed, and then they are going to be attached to an MHC type class 2 protein or molecule, and then that's going to move to the surface, move to the plasma membrane, and be embedded in the plasma membrane like we saw in that first image. So then this cell here, this phagocytic cell, the dendritic cell, has this MHC class 2 molecule embedded on the surface of its plasma membrane, and it also has a little tiny piece of the antigen, so it has an epitope on there. So the MHC class 2 molecule is presenting this antigen presenting cell. They use the MHC class 2 protein to do this. Once this is done, then our T cells are able to recognize this presented antigen and then become activated due to it, which we're going to talk about later. So in this case, what we're doing, basically this dendritic cell or phagocytic cell is going to take the bad guy, break it up into pieces, and say, okay, I need to alert the rest of the body. I need to tell the rest of the immune system what this person looks like, what this bad guy looks like, what this bacteria looks like. So we're going to take the things that most look like this bacteria or most look like this pathogen, and what we're going to do is we're going to hold it up really high. I'm holding my arm up, and I'm holding on to this piece of this pathogen to say, look, everybody, look at me. This is what it looks like. This is what we need to create different things to fight these guys that look just like this. So it's presenting it. It's saying, hey, look over here. This is what the pathogen looks like. This is what we're working on fighting right now. And so this is our antigen presenting cells. So our phagocytic cells do it, so the macrophages and the dendritic cells. And then the B cell doesn't bring it into the cell itself, but what it does is presents this on the MHC class 2, presents a piece of the antigen. And we'll talk more specifically about that later. And then once it is presented in that way, saying, hey guys, look at this, then our T cells are going to kind of come along. And remember, our T cells are cellular immunity, and those are going to connect our innate nonspecific immune response, like these cells, right? The antigen presenting cells, the macrophages and dendrites. And they're going to connect that, not dendrites, dendritic cells, and connect that to our adaptive immune response, right? So it's going to move that from our phagocytes to our adaptive immune system cells. So then when we look at our MHC class 1 molecules, what we see here is that, remember, our MHC class 1 molecules are on every single nucleated cell, so not the red blood cell. The reason that we have MHC class 1 molecules is not only for when our immune system cells are going along the body, they go along the body, they see the MHC class 1 and they say, okay, this is a self cell. Not only are they saying, okay, connected, I see this is a self cell, we're going to move on, they're also seeing if something is within this MHC class 1 molecule. Meaning, is it presenting an antigen piece? Is it presenting an epitope? So the way that this works, the MHC class 1 molecule is going to present an epitope just like the MHC class 2, except it's not only with our macrophages or dendritic cells or our B cells. In this case, it can be any cell in the body, with the exception of the red blood cell. And this is because any cell in the body can become infected with an intracellular pathogen. So this is a way for a cell itself that has been infected to say, hey guys, this is what the bad guy looks like and I'm infected with it. So you guys need to kill me because I'm infected with this. But it's also letting the immune system know what it looks like so that they can go around and start making things to fight these guys off. So the cell becomes infected with an intracellular pathogen. Then antigens that are specific to the pathogen are going to be processed. So again, the cell is going to be taking little pieces as these bacteria or viruses or whatever they are making more of themselves within our cell. Our cell is going to take a piece of the antigen and it's going to then bind it with the MHC class 1 molecule for presentation on the cell surface. So it already has the MHC class 1 molecules up there saying I'm self, like don't kill me, I'm self. But then once it becomes infected with a pathogen that's inside of the cell, intracellular pathogen, then it can take a piece of that pathogen and then put it on that MHC class 1 molecule and then say, hey, this is what I'm infected with. There's a bad guy in here and this is what it looks like. So then that infected cell is targeted for destruction. along with the pathogen, right? Because it's saying like, hey, sorry, we got to sacrifice this cell here because it's infected. It's infected with the pathogen. It's showing us what the pathogen looks like, which is fantastic because then we can build up an immune system response to try to get rid of that pathogen. But it also is telling those cells, okay, this one's infected with this pathogen. Now we have to destroy the cell. And that's why all of the cells in our body, with the exception of the red blood cell, have this MHC class 1 molecule on it, is because not only does it indicate that itself, but it can also indicate if it is infected with an intracellular pathogen. And then remember, the MHC class 2 molecules are only found on our macrophages, dendritic cells, and our B cells, and that's because those are cells that are going to be processing bad guys, engulfing, breaking them up, and then presenting a piece of that pathogen on the surface to then activate other parts of the immune system specifically. So it's not saying, hey, like I've got this bacteria in here, you need to destroy me or break me down, because it doesn't need that, right? The macrophage is fine. It broke down the pathogen and it can now continue on in the body, killing more bacteria, gobbling them up and breaking them down and killing more bacteria. So we don't want to activate apoptosis. We don't want to activate that cell's destruction because it's fine and it's just letting other cells know what the bad guy looks like. So we have the use of the two different MHC complexes, the major histocompatibility complexes 1 and 2.